We were on page two,
Page two.
and identifying members of clades. Basically, there is something we need to focus. Basically, identification of clades is something like we always follow different strategies. First of all, we follow a very important natural feature that is that would be present in similar groups, just like we mentioned that if we find the clause in an organism that looks like a reptile, then we'll compare the structure of the clause, maybe some other things, to classify it properly with the reptiles. So those if we find a new species, then we used to do that particular practice, but that practice got changed. How? We started focusing on the DNA sequences. This basic technique is known as genomics or proteomics, you can say.
Okay.
One kind of it is called as genomics. Wait up, I think okay, sometimes when you test the genes only, it is called as genomics. But
Genomics.
the other times when we study the amino acid sequences in the proteins, it is called as proteomics. My pens
Okay.
have just stopped working. Better. Yeah, okay. One thing is genomics. Genomics is when you analyze the DNA, the RNA sequences, and many other things, and you get to the point that they maybe are genetically similar or different. So that is called as genomics. But when it comes to the other terminology, proteomics, we analyze different proteins and then we understand that whether they are close enough or not. Let's say we have been using the genomics. How? You know the cluster X.
Yes.
Remember that software we used to put the gene sequences into it, and then we used to do what? We used to put the nucleotides, and then we used to compare it. Why did we use to compare it? Because that comparison would help us, or that comparison helps us in identifying the organisms that may be far apart from each other, with reference to the DNA differences, or like you can say the protein. There are other softwares who measure the protein differences. So we can say that we have different kinds of softwares to do that. Anyways, so first thing they identify is the mutation. Okay,
Mutation.
that is something we often do. Molecular clocks are basically based upon the analysis of mutation. Now, what is this? The mutation basically is supposed to occur at regular intervals in humans and relevant organisms. What I'm trying to say over here is that the organisms, the DNA of the organisms performs or you can say, brings a mutation in what I should say, in a very specific time based order. Like, let's say there is something written over here. Um, if I find it out, they are mentioning that the mutations accumulate gradually over long periods of times, and there is evidence that mutation occur roughly constant rate, that they can be used as molecular clocks. Like, we can say that if the mutations occur in human being, one mutation occur in human beings every 100 years. So we can say that we can determine that if each mutation occurs at 100 years, then this mutation might have been occurred 100 years ago, this might have been occurring in 200 years ago, or something like that. Let's say they are putting an example. Mitochondrial DNA from three humans and four related primates have been completely sequenced. From the difference in basic sequence, a hypothetical ancestry has been constructed.
Okay.
Like, you can say that we can use the mutation rate in mitochondria in mitochondria, and we have a set pattern. Like we have a hypothetical value that this number of year, the mitochondria would be having a mutation. Let me show you how many mutations the mitochondria get. This is written in A level book. Let me show you. It is mentioned over there. In I guess it would be in evolution. I guess that's very well observation, selection and evolution, and identifying an evolutionary relationship. Yes, it is over here. Let's say since the mitochondrial DNA is circular and therefore cannot undergo any form of crossing over, changes in nucleotide sequence can arise only by mutation. This is an important statement. Why do we have to use the mitochondrial DNA to create the relationship between those organisms that are mentioned over there? Let's say European, Japanese, African, common chimpanzee, pygmy chimpanzee, gorilla, orangutan. So these this analysis have been done on the basis of the mitochondrial DNA. And why so? Because mitochondrial DNA does not perform any sort of crossing over. Why?
Yes.
Because there is no diploidy. The mitochondrial DNA is only haploid. So
Haploid.
no crossing over occurs. If you remember, I told you that crossing over would occur between the homologous chromosomes. The non-sister
Yes.
homology only appears when you have a diploid set. So no crossing over in any haploid set. Let's get back to that statement. Mitochondrial DNA mutates faster than nuclear, acquiring one mutation every 25,000 years. This is something they have analyzed from some sort of method. Let's say, let's say that the mutation, let's say that the mutation is occurring every 25,000 years. So if we get two mutations, if we get the difference of, I would like see that difference of nucleotides, difference of nucleotides. Okay, let's say between the European man and orangutan, let's say you may have the difference of total different nucleotide as 10.
Okay.
Like how the difference in nucleotides appear. Do you remember?
How different nucleotides appear. That was through a gene mutation.
Mutation. It means if there are 10 different nucleotides between the orangutan and European man, it means that they appeared almost 25,000 into 10 would be,
20,
uh,
250.
yeah, 250,000.
Okay.
250,000 years back, the European and orangutans, you can say that they have a difference of between 250,000. You can say that. Or like this is how the more difference of nucleotides you find in the mitochondrial DNA, the more uh, the farther would be the ancestral distance. Let's say there is only the difference of two nucleotides between the European man and the Japanese man. So what does it
Yes.
mean? When did they split? They split it up almost. If you got two mutations between them or difference of two nucleotides, what does it mean? One mutation occur in 25,000 years. So
Okay.
two mutation would be in 50,000 years. It means
50,000
that
years.
they have been apart from each other around they divided or they separated from each other around 50,000 years ago. This
Okay.
is what the meaning of drawing the clade according to the mitochondrial or genomics.
Yes.
For according to the mitochondrial DNA or genomics, let's see. 70,000 years ago, European and Japanese split. We were almost close, by the way.
Yes.
We were almost close, which means that maybe there may be three mutations, almost three mutations are there, because 70,000 means 25 into 3, that would be 75,000. So,
Yes.
but almost three mutations, the difference of three nucleotides existing between them, it means that They are like we can analyze their difference by, and we can create a cladogram. This is a cladogram. This is a
Okay.
cladogram. So
Clever.
we can create a cladogram on the different on the basis of base differences between them. Then 1,40,000 years ago, 1,40,000 means like uh, 25,000 into 5 would be 1,50,000, 150,000,
150,000.
150,000. So it means that they found almost between 4 to 5 mutations or base differences between African, European, and Japanese split.
Okay.
They are discussing this whole particular group. Then it's I guess 5 millions. 5 million. So 5 million years ago, human chimpanzee split. That is like, where is the human if we consider this is the human? Basically, you can say this is the lineage of human.
Yes.
This is the lineage of human, and this is the chimp. So basically, when did they split? You can say they got split from around here. This means 5 million. This is 5 million. So this occurred 5 million years ago, and they got split. Did you get it?
Yes, sir. The 5 million. It's up to us. Like, how did you know it's exactly 5 million, the value?
We That's a really great question. We actually don't only perform the mutation analysis or nucleotide difference. We don't always conclude on the basis of we don't always conclude on the basis of mutation rate. We also perform the dating procedure. We also perform different kinds of fossil analysis and other things, and we can analyze that. You can say that we adjust our analysis according to multiple opinions, not only on the basis of mutations or mutation rate.
Okay.
Got
And
it?
yes, sir.
Hmm.
And this might be a silly question, but humans, right? We're saying that the mutation rate is almost once every 25,000 years, right?
In mitochondrial DNA, yeah.
In mitochondrial DNA.
Hmm.
Okay. So for humans in this case, would we not expect the humans to change like quite a lot with the mitochondrial DNA? Because human Homo sapiens, they've been along for almost like 200,000 years, right?
Yeah, and the mitochondrial DNA can show some mutations, but you know what the problem is? The problem is that, let's say, if we create a cladogram, where was your board? This one, I guess. Okay, if you create a cladogram, you are going to get the human cladogram, like, let's say, because on the basis of DNA, if you like create a cladogram on the basis of genomics, then remember that the DNA dies within, let's say, 10,000 years. The DNA finishes.
Okay.
It is too much stable, and it remains there for a long time, but it it is dead after a few years, around 10,000 years. Like, it means that if you want to analyze the if you want to analyze the mitochondrial differences between the human beings, then you don't even have the complete data that what was the nucleotide sequence of humans before 10,000 years. And
Okay.
like
We're saying the
how.
nucleotide. Oh, sorry,
Hmm.
sir.
Yeah, please, 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 please.
Okay. We're saying you said the nucleotide sequence it gets deleted after 10,000 years.
No, 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 DNA gets denatured. It
DNA
gets
is just
broken. It gets broken, I guess, maybe 10,000 years, or we can write it on the Google. The for how long a DNA molecule can exist in a fossil. Hmm, how long does DNA last in fossils? Okay, 1.5 million years. No, my analysis was too less. 1.5 million, 1 to 1.5. Wait up. How long does DNA molecule last? Leg bones of extract MOA birds in New Zealand found that half life of DNA is 521 years. So every 1,000 years, 75% of the genetic DNA information is lost. Every single base pair is lost. Like you can say that what point they are making. DNA has now this is like confirmed. You cannot reject this one because this is nature. Have I told you about the nature?
Yes, you said the Nature was a really good website.
Yeah. So if they are saying 521 years of half life, it means that every single nucleotide would be lost in 681. But the problem is, we need the DNA for analysis. We
Yes.
need the DNA for analysis. We don't need one single nucleotide. Let's say we found a nucleotide, but what is the use of it? If we don't find a proper sequence, then there is no use of it. If you find very small pieces of DNA in a fossil, you cannot even join them properly. You cannot even join them properly. Let's say, let me write it down. We found some of the fossils in the mammoth. You know the mammoth?
Yes.
Hmm. Uh,
Elephant with the really
age
long
of
horns.
yeah, age of uh, fossils of mammoth. 5 million years ago. 5 million years ago means that and like you can say, we can write another question. Have we found the DNA in any fossil, or we can say DNA, the intact DNA in any fossil? The oldest DNA found and extracted from mammoth specimen that was potentially up to 1.6 million. It means that we can find the sequences. That is like the oldest DNA that we have found is 1.6 million, which means 160,000.
Yes.
So my my figure was like too less. I guess it was something else, about something else. But the thing that you were asking that why the human is not getting mutated.
Yes.
Okay, the problem is, uh, we know the human, we know the human DNA for how long, by the way?
The Homo sapien, I think so. That's
Yeah.
almost 300,000 years old, right? Or
Uh, have we found 300,000? Okay, the Homo sapiens 300,000. So is it possible that we could have found the DNA in the 300,000 old fossil, years old fossil? According to our information, we we could not find any DNA from any fossil older than 1.6 million years old fossil. Older
yeah.
than 1.6 million. What does it mean? That if we find a fossil older than that, then like almost, well, we found no human DNA. And still, if I write it down, let's say, uh, we haven't found any intact human DNA, by the way. Have you found the intact DNA of in any fossil of genus Homo? Okay, what is the fossil evidence of the X woman might be a member of the species Homo erectus? No, wait. 18,000 years. Okay, the oldest sequenced DNA from a modern human extracted from 45,000-year-old bones in the caves of Bulgaria. Now, the thing is, now I'm about to complete your answer. So much time has been passed. The
Yes.
reality is that uh, if we try to analyze the mutation in human, we may find some of the nucleotide differences in human mitochondrial DNA, depending on what DNA did you find. Like, let's say you may found the nuclear DNA, but you don't know the clock of mutations of nuclear DNA. Like, let's say they found the 45,000 old molecule of DNA, but that DNA was nuclear, not mitochondrial. What does it mean? They don't know the molecular clock for nuclear DNA. What does it mean? That they cannot determine that how many mutations it is having, how many
Yeah.
mutations it is having. But we are talking about the mitochondrial DNA. So always remember, it depends on two things. Number one, intact DNA, two to three things. Number two, it should be from the mitochondria, so that we can find the differences in their differences in their split. And then we should be able to identify, like, we should be able to test it, we should be able to sequence it. If we find the DNA but we cannot sequence it, what is the use? We don't have any use of it.
Yes.
Like, I would, again, I would say that we physically only know the human. Like, how long do you think, like, how many years of history of human do we have?
Uh, that we have around maybe to like the 1300s, right? Before that, we don't have so much history or any like fossil evidence.
Yes, 1300. What you are saying, 1300?
Yeah, around 1300s.
Okay. And all right, you can also say that, like, maybe you have a history of human beings, like, you may know something even before Christ. You know the
Yeah.
Shakespeare, you know the Aristotle, you know the there was another one, Aristotle, and what was the other one? Okay, let's leave it. All the great people, before Christ even, like, how long they've been dead, like, what is the, like, total number of years, you can say, you can count after their death till now. Like, maybe someone may survive 5,000 before, someone was maybe living 5,000 years before Christ, maybe. But what is the meaning of before Christ? 5,000 years before Christ mean the total years till now are 7,023.
7023.
Because before Christ are 5,000, and, like, if you count these 80 years, so we are in 2023.
Yes.
So just add them: 5,000 before Christ, 2,000 years after Christ.
Okay, yeah,
So
73,000 around. Yes.
yeah. So the thing is that the total number of human beings and their characters, we barely know them. Why? Because 7,000 years is no large clock for the evolutionary, from the evolutionary point of view. It just it's merely the blink of an eye.
Okay.
So if you try to find the difference in the characteristics of human being, it is a rare event that you would find the intact DNA. And if you did find it, maybe it's not mitochondrial, so you would not be able to do a perfect analysis on the basis of molecular clock. And number third is, if you have found the intact DNA, are you able to sequence it? Like, in modern times, yeah, maybe, but there is no report about it yet. So you can say that it's quite a difficult procedure.
Okay.
They have actually they have actually drawn a cladogram over here. I'll show you this. I'll explain it. Let me pick a call.
Okay.
So I was saying that this is a cladogram they have mentioned over here. Now, these are basically different types of lizards, but you can see that they have put the sphinodon and heloderma m- farther away from each other compared to Agama and Cameleo and Opplurus and Anolis. Why? Because according to the molecular clock, they found that there are lesser differences in nucleotides of these lizards compared to these ones.
Yes.
So they created a cladogram according to this.
Okay.
Then analogous and homologous traits, we have talked about this much already.
So
Um, yeah, please.
in the case of the lizard, where there's such a big difference, that is an example of the punctuative equilibrium, right? Because scientists like, according to evolution, there should be some intermediate between the sphenodon and agama, right?
Exactly, exactly.
Okay. Okay, sir.
Okay. Now, getting to the homologous and analogous characteristics, we know that homologous are those which are different in functions, but their anatomy is similar, and
Yes.
because of same ancestry. And analogous are those who may have the same functions, but their anatomy is different, and that is because of uh, uh, separate ancestry.
Okay.
So, do you do you need an explanation for that, or is it enough?
No, I remember. I remember we discussed the analogous structures, for example, wings, fly, and the bird. It's
Yeah,
different,
exactly.
the same function.
Hmm.
Okay.
Oh, all right. Now, getting to these are basically the eye structures of the octopus and the human. They are strikingly similar, and that is like something very much strange. Now, these are tree diagrams that show most probable sequence of divergence in clades. What is a divergence? Divergence means that how much they got separated. So, basically, in modern times, the cladograms are made on the basis of made by the computer. What do we add in the computer? We add the amount of differences in nucleotide sequence and amino acid sequences and base sequences. Like we discuss the proteomics and genomics, and then we determine that how much difference would be there. So
Yes.
actually, it is the computer that creates a cladogram, that
Okay.
creates the cladogram. So uh, the important part of this is this picture, very beautiful one. Just look at this. This is the ancestral species C, and some group got separated from this branch, and the rest of these are like on this way. So
Yes.
lizards and snakes are there, and the rest of it are like these are the ancestral species for birds and mammals, maybe,
Okay.
because like maybe this ancestral species B is for crocodiles, and the ancestral species A is for dinosaurs, non-avian birds, and birds. And then maybe someone here would also be an ancestral species for the mammals.
Okay.
All right. The branching points are called nodes. Yeah, two clades branch off. This is actually a dichotomous tree, by the way,
Okay.
because
Dichotomous.
it shows the dichotomy. It splits into two, as I
Yes.
mentioned. So it splits into two on the basis of one, two, or more characteristics. So it also looks like a dichotomous key. Anyways, then comes the this is the explanation of cladogram. Birds, non-avian dinosaurs, and ancestral species A from clade called Dinosauria. Then birds, non-avian dinosaur, crocodiles, and ancestral species are part of a clade called as archosaurs. So they have named different clades, and they have different names for different groups here. But remember, they all are these are clades, and they all are also clades. It depends on that what ancestor you are considering.
Yes.
If you are considering a close ancestor, the clade would be smaller, and vice
Okay.
versa. The primate cladogram, they are telling us that the number of on the cladogram are estimates of the population size. Let's say the bonobo, chimpanzee, and human. Their ancestor had 45,000 of individuals, out of which 27,000 were got separated 4.5 million years ago, and the rest of it, out of 25,000 were 27,000, it would be about um, 18,000. Yeah,
Yeah.
18,000, and out of these 27,000, they got separated, maybe due to sympatric or allopatric speciation, and the 12,000 were here, and the rest of them were in chimpanzee group, and same for the human.
Okay.
So, like, they can find out the fossils, and they can find out that how much they of those organisms would be present over here. Okay, analysis of a cladogram. Analysis to deduce evolution. I have been explaining this lately
Yes.
based on the mitochondrial and DNA and proteomics. Because genetic changes are assumed to occur at relatively constant rate, these numbers can be used to estimate how long ago two clades diverged. It is
Yeah.
called as a molecular clock. So, if we know the molecular clock, remember, if we know the molecular clock, only then we would be able to say that how long ago they got separated. If we don't have a molecular clock, we would not be able to understand that how long they have been living as a separate species.
Okay.
They cannot be regarded as a proof. Cladograms are constructed on the assumption that the smallest possible number of mutations occur to account for current base or amino acid sequence differences. This assumption is incorrect, and pathways of evolution were more convoluted. It is therefore important to be cautious in analysis of cladogram, and where possible, compare several versions. Like, sometimes you find similarity, but that similarity is not in two closer organisms, just like the birds and bats.
Yes.
Like, you find similarity in gene sequences, or maybe you find similarity in the, I would say, you find similarity in the protein amino acid sequences. You can find it, but it is not necessary that it is all time. Always, it is true for the cladogram to be corrected. What they are saying, constructed on the assumption that the smallest possible number of mutations occurred to account for current base or amino acid sequence differences. Sometimes this assumption is incorrect, and pathways of evolution were more convoluted, which means that maybe it is not that much right. It is not that much right, because if we conclude all of our evolution on the basis of only this genetic or protein sequences, maybe we are wrong. Maybe the difference is not due to mutations, or the common genetic sequence is not due to common ancestor. Maybe it is because of some other reason. Are you getting it? Like,
Yes.
if you find the same DNA in two different species, it does not mean that they have a common ancestor, but it may have any other reason, and you have to consider the rest of the characters as well.
Okay. So in the case of the bird and the bat,
Hmm.
the code for the wings, would that be the same in both cases, or they'd be quite similar, right?
Quite similar would be the proper statement.
Okay. But even though that they're quite similar, there are
Hmm.
other, like for example, the beak and the bat's, vision and other stuff,
Yeah.
they're completely different. So that's why we can't assume that they're the same in the same
Hmm.
cladogram.
Let's read this. A cladogram for the great apes. The great apes are a family of primates, taxonomically and hominidae. There are five species on Earth today, all of which are decreasing in number apart from humans. Cladogram for three of the species. Use this information to expand the cladogram include all great apes. Okay, figure 6.
Six.
You may use it for that. Well, let's move towards the database question. Cladograms based on morphology suggest that turtles and lizards are not a clade. They are not a clade, which means that maybe they are not sharing the same ancestor, what they want to say. To test this hypothesis, microRNA genes have been compared for 9 species of chordates. The results, chordates are like vertebrates. The results were
Okay.
used to construct the cladogram in figure 8. Construct the cladogram. The number on the cladogram show which microRNA genes are shared by members of a clade. but not members of other clades. For example, six microRNA genes found in humans and short-tailed opossums, but not in any of other chordates. Deduce using evidence from the cladogram whether humans are more closely related to the short-tailed opossum or to the duck-billed platypus. Calculate how many microRNA genes are bound in the mammal clade are found in the mammal clade on cladogram, but not discuss whether the evidence in the cladogram supports the hypothesis that turtles and lizards are not a clade. Evaluate the traditional classification of tetrapod chordates into amphibians, reptiles, birds, and mammals. All right. So we can conclude all of this by this particular diagram. I would
Yes.
suggest you to do this, like practice it, so you
Okay.
can create the conclusion. Because like we can understand many things, and I have told you many things already. So you
Yes.
can analyze it, and you can come up with an answer. The important thing about the answer that it should be relevant to the question. If you are if you successfully complete these questions in the final examination, your answers would be based on the proper information from the book, not from your own mind, or not from your beating around the bush. So it would be from the book. It would be better answer.
Okay.
So uh, it would not look like you don't know the answer and you are just trying to fill up the space.
Okay, sir.
Now, evidence from cladistics has shown that the classification of some group based on the structure did not correspond with the evolutionary origin of. This is what I was talking about yesterday. That the cladistics, those scientists who had a cladistic approach, they when they started classifying the organisms on the basis of clades, like the ancestral basis only, then they found that putting two organisms in the same group or in the same taxon is not suitable, because sometimes their characters may look similar but they belong to different ancestry. They belong to different ancestry. Cladistics, cladistics has caused some revolution in plants and animals. It now clear from the cladograms that traditional classification based on morphology does not always match the evolutionary origins of the great groups of species. Some groups have been reclassified, and I guess I did not tell you, but um, you know that there was a kingdom Protoctista. Did I tell you?
No, not protectista.
Okay, then
Oh,
just
you
leave.
might have told me. You told me that there was initially, I think so, six kingdoms, but we took
Five.
away one. Five
Yeah,
kingdoms.
that is the
Yes.
thing I want to mention over here. That due to the claristic approach and particular evolutionary relationships, they removed the kingdom Protoctista. Why?
Okay.
Because that kingdom was not actually following all the rules. So they had to change it. Now they have created five or six different groups for that group, but not a single kingdom. So that
Yes.
is why, like, sometime it creates many problems. But yes, you can reclassify every time you see that a particular organism should not be placed in group A. Maybe it belongs to group B based on the ancestral relationship.
Okay.
All right, now we have cladograms and falsification. Uh, with one theory being superseded by another. The reclassification of plants on the basis of discoveries in Claris is a good example of an important piece of the testing of theories. The classification of angiospermophyte into families based on their morphology was begun. Now, what they are actually saying that what is falsification? Falsification means that whenever you find something new, you try to reject the older one. So
Yes.
it is a method, and the scientists use it. Although it helped them a lot in classifying the organisms that were previously very much difficult to classify, but they put them in the proper groups according to their particular ancestral features.
Okay.
All right, then classification of the figwort family.
Figured family.
Okay, what they are saying, what they say, he gave it to us. As more plants were discovered, the family grew until there were 275 genera with more than 500. That is quite a lot. Like
Yeah.
20 to 75 different genera. That is quite a lot. Okay, transition recently. Taxonomists recently investigated the evolutionary origins of the figwort family. One important research product compared the base sequence of three chloroplast genes in a large number of species in genera traditionally assigned to the uh, Scrophulariaceae and genera included. It was found that species in figwort family were not a true clade and that five clades had incorrectly been combined into one family. Now, like two were proper and five were improper. So this is like something happens when you try putting them in the groups that do not share the common ancestry. So
Yes.
that basically is going to be the result that you have to rearrange everything. That is why the cladistic approach or the cladistic whatever you say like the cladistic approach or maybe the evolutionary approach, the phylogenetic approach or taxonomical approach, they all have to work together to think together so that they can come up with a whole new classification through from which you cannot like from a new classification from which they would take help that if a new species is discovered they would it would become much easier for a particular for the classification of the new organism that has been just that have been discovered just yet.
Okay.
All right. Okay, it is more than 36th largest among the angiosperms. Okay, this is something the example. Like if you want to, you can. These are the questions. Try doing this first. Try doing the exercise first.
Okay.
When you would be able to do the exercise, then it would be much easier for you to solve the paper-based questions or the worksheets.
Okay, sir.
Any particular question?
So far it's clear, and the reason for reclassification of clades, it's that it's not that like the genetic information is not similar. It's just that the
Hmm.
ancestral linkage it does not, you could say, it does not make sense in a way. It does not line up.
Hmm.
Okay.
Like their ancestors may be far apart. So if they are far apart, you cannot consider them behaving like each other. Let's say I would mention the example of a bat again. The bat does not complete its attack until it like almost kills it or hurts it badly.
Yes.
But on the other hand, the birds have different way to kill their prey or like they have totally different types of food. So that is something different. What I was saying actually, you know what
Ye-
happened? I just got slept. Oh God,
okay.
I just slept during the class. I did not know what I was saying.
Okay, sir.
For God's sake. Okay, trying to refresh. All right. Literally, I was speaking the words, but I just it just flashed off my mind what I was saying about the bats attacking on their prey. What was I saying? Something like that.
Yes, sir. You're saying that the bats attack their, it was like attack their prey till like they're like really badly injured. Whereas the birds, they have like a different hunting method, and that's like a difference in clades, you could say, because the ancestors.
Okay, for God's sake. Okay, okay, fine. Oh, thank God I did not say something silly. Okay, all right. So, I'm trying to sit properly. All right. So, uh, there has been like a major classification has been carried out. Less than half of the species have been retained in the family. Now, this is something they are trying to say that they everything now has been under consideration. Like the scientists are reconsidering all the things, all the stuff, and they are just realigning their classification so that something should be. You can say some at some point all of them should be agreed. The taxonomists, the phylogeneticists, and the cladistics, they should be agreeing on some common point so that the classification may reach to a perfection point. But it cannot be obviously done because since you have already been mentioning a point repetitively, and that was the punctuated equilibrium.
Yeah.
In the punctuated, since we believe, or like if we believe in the approach of punctuated equilibrium, I would say that uh, m- they would not be able to agree on one point, because if you start ignoring the missing links, then you would never be able to agree on a single point. uh, like whatever approach you use, taxonomy, cladistic, or phylogenetic, whatever approach you use, if you try to ignore and bring on the punctuated equilibrium point, then maybe they would never be able to classify all of them.
Okay.
All right, I did say something about the exercise, right?
Yes,
Did I ask you to do it
you said to
first?
do these exercises and then attempt the what's it
Yeah.
called? Past paper questions.
Yeah, that is actually why I said that. Actually, I have been telling my A level students lately that start doing the exercise questions, because it has been recently followed, or maybe not that much recently, but since like almost 5 years, they follow a trend that they put the past paper questions in the exercises. So what happens, like if you do if you follow the exercise, you would have a lot of experience of, like you can say, collecting the words, doing the exercise. Doing the exercises would make you in practice, would keep you in practice. Like you would be practicing the questions if from the exercise as well as the past papers, so it would be better for you.
Okay.
All right, any question, any particular problem?
It's clear so far, sir. I can maybe also send you the questions the school gave on the what's it called? The WhatsApp group for chapter 5. Maybe
Sure.
if those questions are also similar to the book.
Sure. Another thing I wanted to ask. I forgot, when is your exam?
My exam, I have the date. It's around. It's November 3rd or 4th, sir.
3rd or 4th November, and it is September.
Yes.
So it's almost mid of September. So like we have done genetics, we have done evolution and biodiversity, we have done have we done the nucleic acid?
Yes, sir.
Yeah,
You can take guesses.
we have done the genetics and evolution. So far, we done much of it. This one is much easier.
Okay, sir.
Okay, metabolism. What about this chapter? Do you have confidence in this chapter?
Yes, this chapter was quite short, sir. It was quite
Hmm.
close to chapter 2 as well. It has quite a
Yeah.
lot of points there.
Mm-hmm. Okay, fine. What about chapter two?
Chapter 2, I had to cover because it's really important. Oh, I've covered it. It's the DNA structure, enzymes, water molecule, lipids, uh,
Yeah.
all those basic. It's basically all has the basic concepts that the later chapters you need to know.
Cell respiration and photosynthesis is also given in the here. So
Yes.
obviously it is that was for SL and this is for HL. Okay. All right. So, like, once we are done with, let's say, ecology and okay, we are left with the physiology, and you were weak in physiology, right?
I physiology, sir. Physiology, I don't think so. I think so is the option mostly.
Okay, in optional animal physiology, you actually are like lacking.
Yeah.
Or okay, it is human physiology, then animal physiology. It is a part of HL. So
Yes.
that this physiology, human
Yes. The
physiology.
same is option D.
Okay, okay. Well, that's not that much difficult if I remember from human nutrition digestion. Anyways, some people may find it difficult, no problem. So,
No, I just
or
thought
more?
because it was quite a lot with the heart, memorizing where the blood flows, the shape of the heart. I thought it was
That is
quite like a lot to memorize and understand
mm.
and grasp.
That's right. Like that, it has a lot to memorize, and that is something I would say. Like we don't need to memorize the thing, but we still need to memorize. What is the meaning of my sentence? That like according to the theme of IB, we don't need to memorize it, but when we appear in the exam, it appears that we have to memorize, although they say that we don't have to, because
Yes,
like many points, like if I remember the labelings of the heart, you don't memorize it, you can never take a hold on that topic.
yes, sir.
Anyways, let's start. All right, okay. Now first of all, tell me, what do you know about the ecology?
Ecology, you could say it's the studies of how species interact in certain environments.
Hmm,
And
okay.
in ecology, you study about the species and the different ecosystems, and you also discuss about carbon cycling and the climate change.
All right, what is an ecosystem?
An ecosystem, you could say, an ecosystem is where a group of population or a community in a specific environment, like a natural habitat, right?
Hmm. Okay. Now, the ecosystem, basically, it's like if I try to start from the basics, the word ecos, or maybe it's ecois, ECOES, I guess, OES. It means home
Home. Okay.
or household, and logos, study. So, ecosystem basically is a particular area where the abiotic factors factors have, or I would rewrite it. Wait up. If you were writing it, I'm sorry. Uh, a group of organisms belonging to different species belonging to different species, biotic factors. That would be the biotic factors. Interact with each other and their environment and their environment. So that is something we say abiotic.
Abiotic.
Okay, always remember that the ecosystem basically designed it is basically designed on the quality of interactions, quality and quantity. Like, if you want to know about the ecosystem, if you want to know about the health of an ecosystem, if you want to know about how much an ecosystem is diverse, just look for the interactions that exist in them, the
Okay.
quality wise and the quantity wise. Like how many interactions are existing and how many organisms are following the interaction. That would be the quantity, and the first one would be the quality. So first of all, always remember. Let's try to split this definition. I'm not following the book right now. Let's
Okay.
say a group of organisms belonging to different species. Now, this, if you if you want to put this whole sentence into one word, group of organisms belonging to different species, different species. If you put it into one word, that is called as
Community.
community. Remember that a community is an ultimate requirement for an ecosystem to exist. Why is that? Because it is a community in which the interactions exist. Why? Interactions are possible in the community. Why so? Uh, let me provide you another terminology. There is a terminology called as population, in which you say that it is a group of organisms, group of same organisms, and which means they belong to same species.
Yes.
They live at the same place and same time. What does it mean? It means that a group of 20 lions may be a population, and maybe the lions may live somewhere else as well, but that would be a different population. Let's say in my in the society, in the housing society where I live, maybe the total number of people is 5,000, let's say.
Yes.
So it is the population of this society. If you compare it with another society, their population would be totally different. What does it mean? That it is a bunch of an organism which are same in which are like they belong to same species, and but they are living at the same place at same time, and they are separated from any other such population. If a
Okay.
population is living 10 kilometers away from me, they are Homo sapiens, I am Homo sapiens, but I have my own population and they have their own population. So the population always consists of the organisms belonging to same species. Now,
Yes.
when it comes to the population, always remember that the quality of relationship is compromised, which means very less types of relationships, very less types of relationships. Now, what it literally means, it literally means that if you expect a population to have relationships, that would never be building up so much relationships, and the only relationship that would be existing within a population is a competition.
Competition.
Okay,
Okay.
to understand all of it, first of all, let me give you a random definition of a relationship in an ecosystem. A relationship is when two organisms live together, live together, and have an impact upon each other's
Life.
life. That is what we often call as the relationship. So,
Okay.
like if two individuals from the same species are living together, how do you suppose they are in a relationship?
They would probably be in the same, what's it called? Community, right?
No, no, no.
Because
I'm talking about a population. If two individuals are living together, belonging to same species, they
yes.
are living together, but they what kind of relationship would exist between them? That would be the competition. Why?
Competition.
Because if they belong to same species, it means they would compete for food, as well as they would compete for mate
Yes.
or their partner. Now, that is what I'm trying to say over here. That if they live together, They would have a relationship, but the quality of relationship would only be a single type, and that is the competition. And this is not an healthy ecosystem. Why? The healthy ecosystem would have a lot of individuals and a lot of relationships in them, types of relationships in them. So the only relationship in a population is the competition. When it comes to the community, when it comes to the community, the quality and quantity of relationship increases. The quality and quantity of relationships increases. How? When it comes to a community, there is a type of relationship called as parasitism.
Parasitic.
Then comes the mutualism. Mutualism. Then comes, like, let's say, commensalism. There is another word. Maybe I would not explain all of these, but what I want to say over here, that it increases the chances of organisms interacting each other, having an impact on the lives of other partner. So
Yes.
more, what does it mean? More the relationships, healthier would be the ecosystem.
Okay.
Healthier would be the ecosystem. So
Ecosystem.
this is what the meaning of relationships and interaction means. Erection, like the interactions, they are basically, I would say that they are basically studied in the community, not in the population.
Okay.
So number one, group of organisms belonging to different species. What does it mean? Ecosystem is defined by a community, because different species only exist in a community, not in a population. What occurs in a population? In a population, are there different kinds of species?
No, there's the same species.
Yep. Now, coming to the biotic factors. What is the meaning of biotic factors?
Abiotic factor. This I don't know, sir. Abiotic.
Just try to define biotic.
Okay.
You don't know?
Biotic, you could say biotic is in environment, right? Or
No.
it's, no?
Biotic is all the living components of a community,
Okay.
components of an ecosystem. Like, let's say, if I am studying a community, then all the living organisms would be considered as biotic factor
Biotic.
of that particular ecosystem, and they would influence each other's life. And what are the abiotic? It is pronounced as abiotic. Abiotic is
The
all
non-living.
non-living,
Okay.
non-living parts. Now, how you would consider all non-living? Let's say pH, temperature, wind speed, soil quality, climate, season, whatever it is, that would be that would be an abiotic factor.
Factor. Okay.
So, what is an ecosystem then? Ecosystem is a relationship between the biotic factors. Within them, the interaction also exists, and between the abiotic parts of an area.
Okay. So
Abiotic part.
so we could say the relationship between living organisms and the environment or the abiotic environment.
And the relationship also would exist between the same types of I'm sorry, the relationship would not only exist between the biotic and abiotic factors, but it would also exist within the biotic factors.
Okay.
Like, parasitism is a relationship between two different organisms, then mutualism, then commensalism. There is another relationship that only occurs in a community, and that is a predator prey relationship.
Yes.
Predator prey relationship, and that also occurs. So we would say that predator prey would also occur in a community. So it is important to study the relationships. It is very, very important to study the relationships to understand an ecosystem.
Okay.
Now, you may feel that the very first thing that have been discussed in this chapter is the species. Why? Because species is a smallest group. a small group of organisms. So first of all, we need to understand the species, although we do know, but before studying any ecosystem, we must know that when it comes to ecosystem, the unit of ecosystem is the species.
Okay, unit of ecosystem species.
What does it mean? It means that more the species, stable would be the ecosystem.
Ecosystem.
It's like unit of life is a cell.
Yes.
Unit of life is a cell. More the cells you have, working cells, better you would be working, better you would be surviving. Similarly, the number of species would define the quality of an ecosystem. So, when it comes to the species, but if we talk about only one species, that would represent the population of an ecosystem, not the community. But
Yes.
when you combine many species, when you combine many species in a particular area, then it would become a community. So,
Okay.
and like I said, first of all, species is the smallest unit. Then combining the species would create the community.
Yes.
And combining, when you combine the communities, you would create an ecosystem. And when you combine the ecosystems, you would most probably be getting the biomes.
Biomes.
Biomes. What is the biome? If this word would come in the book, I would explain it. So
Okay.
these are different levels. We study the ecology on the basis of species,
Yes.
and this is called as population ecology. What does it mean? We only study the ecological principles for one species. That is called as population ecology. But when it comes to a community, we study the the ecology for a community. We study the ecology for a community, so that is called as community ecology. And in that ecology, there is a lot of, you can say, that kind of community. If there is community ecology, then you can find a variety of organisms. You can find many organisms to study. You can find many new organisms in a community. So that is
Yes.
mostly when we study the ecology, we keep in mind both factors. We study on the basis of species level, and we also study the community level. So that is what the meaning of the ecology is.
Okay.
uh, the species we have described much, the population. Now there are some qualities of the organism that they are trying to describe, or like you can say that I can ease many things for you. Let's say, like, if I talk about the biotic factors, what did I say about the biotic factors?
The biotic factors. Biotic factor is all. Biotic is living organisms, and abiotic
Hmm.
it is all non-living organisms. So we
So
said
let's say we have types of biotic factors. Okay, now when we classify the biotic factors into groups, then we have a criteria, like how they obtain nutrients. You remember the five kingdom system?
we had autotrophs and heterotrophs, right?
Yeah, autotrophs and heterotrophs. How they obtain nutrients. So, first of all, the very first criteria of the biotic factor is the autotrophs, and the second one is the heterotrophs. And heterotrophs have something. Autotroph obviously are those which are including the plants
Yes.
as well as the planktons. You know about the planktons?
Plankton. It's found at sea, right? The plankton.
Yeah, the
Okay.
planktons basically are unicellular plants. They
Yes.
basically are unicellular plants. Like, they perform the activity of photosynthesis, but they are unicellular. They are not multicellular.
Okay,
Okay, by unicellular and multicellular, I just remember. Did you read the classification topic?
specific. No, unicellular versus multicellular. That's chapter one, right?
No, no, no. The one I taught you yesterday, the classification
Classification. Yes,
table.
the kingdom table. Yes, I went through the notes of how we classify them.
Like everything clear?
Yeah, it was clear. As I told you, I couldn't really find any questions on them to classify, but the book examples, I understood how to classify it.
Now, heterotrophs have many types. Obviously, we know that autotrophs are only plants and planktons, but autotrophs are of many. I'm sorry, heterotrophs are of many types. Autotrophs have been classified. Heterotrophs are of, first of all, consumers. They consume the food. They just eat the food.
Yes,
They don't care of the environment, they don't care of anything else, they just eat the food. So that would be the heterotrophs, and they are meant to take the food from the plants and all the other stuff. Like plants gives the food to to buffalo, and then it would be giving the food to maybe an organism who would attack on the buffalo and kill it and eat it. So the energy or the all the components would be transported to the next organism. All those organisms who are obtaining food from the plants or other sources, they are heterotrophs.
heterotrophs. Yes, I remember
Okay.
they
Mhm.
say the autotrophs. They're always because we refer to them as producers.
Yeah.
Okay,
Okay. In consumers, we then divide them. Like, let's say we have a very particular division for consumers. Like, uh, the autotrophs are also known as producers, autotrophs. But we have consumers. So, what are consumers? As I told you, that consumer is the word for all those organisms who are heterotrophs. Heterotrophs. Okay. All right. We'll do the rest in the next class.
okay. Sir, I just checked the exams is 8th of November and 9th of November. So
Hmm.
almost a bit less than 2 months.
Okay. Okay. Right.
Okay, sir.
All right. Thank you.
Have a nice day.
Nice.